Following injury, muscle stem cells, called satellite cells, give rise to transient amplifying progenitors that will differentiate and fuse to make new myofibers. Our lab, at the Institute of Myology in Paris, is interested in the molecular pathways controlling muscle tissue repair in mice.
In this paper, we studied the role of canonical wind signaling during skeletal muscle regeneration using beta-catenin loss of function or gain of function mutations specifically in adult PAX7 expressing muscle satellite cells. After inducing recombination of the beta-catenin gene by applying tamoxifen to adult mice, we injured the tibialis anterior muscle by injecting cardiotoxin and analyzed the early regeneration process 7 days post-injury.
At this point, in both beta-catenin conditional knockout and beta-catenin conditional active muscles, myofibers were smaller compared to control muscle, as measured by the cross-sectional area, or CSA.
Moreover, the mean number of nuclei per regenerated myofiber in beta-catenin conditional knockout muscle were reduced compared to controls when quantified at 7 days and 38 days post-injury. This suggests a defective differentiation of fusion process in mice with conditional beta-catenin modulation in muscle stem cells. Following conditional beta-catenin activation, the number of myonuclei per fiber was comparable to controls in early stages, but was significantly reduced at the end of the tissue regeneration. This indicates that the myogenic progenitor pool is exhausted in later generation of those mutants.
Next, we quantified the numbers of differentiating muscle progenitors expressing myogenin. Those myogenin-positive myocytes were reduced in number when beta-catenin was knocked out. Accordingly, the number of myogenin-positive myocytes was strongly enhanced in mice expressing the conditional active beta-catenin in muscle. The embryonic forms of myosin-heavy chain marks all newly formed myofibers between 3 and 5 days post-injury and is absent from mature myofibers in controls. We observe a high number of lag myofibers expressing embryonic form in beta-catenin conditional knockout muscle, and thus a delay in the regeneration process. Interestingly, lag myofibers were only seen occasionally when muscle satellite cells expressed conditionally active beta-catenin during regeneration.
From this in vivo data, we conclude that the myoblasts fail to differentiate in the absence of the beta-catenin, and that the conditional activation of the beta-catenin muscle satellite cells pushes myoblasts towards differentiation at the expense of amplification. Supported by the in vitro data of this paper, we suggest that canonical wind signaling controls the balance of the proliferation and differentiation in muscle progenitor cells during tissue repair.